All right, guys. We are very honored to have 10x Genomics today with us. And I'm Jasmine Chen. I'm the Director of Genomics at A Biosciences. And today we will talk about the cytosines that we newly introduced in our company and that our validation. And we will have John first to give a introduction about the technology and what 10x is about to bring, perhaps. And it will follow with a lunch break, likely from 11:45 to 12:35, or maybe we will decide. And then I will talk about our laboratory work on the cytosines validation and our in-house work to showcase our work on the cytosines VCN. And Nasim Ramapour is our bioinformatic scientist, and she will give a presentation on the combination of the VCN work using the single cell to deconvolute the VCN data. So we are very looking forward to this exciting webinar. So John.
Hi, everyone. Looks like my camera isn't working, so if there's no audio, you may want to turn up your volume just to make sure my audio is coming through too. So yeah, my name is John Manuel. I'm the Spatial Science and Technology Advisor. Thank you for joining us today. I will be covering our, give me 1 second here. There we go. All right. All right, yeah, thank you for joining us. I'm John. I'm the Spatial Science and Technology Advisor with 10x Genomics. Thanks for joining us today. I'm going to cover our Visium spatial transcriptomic solution, in addition to covering our CytAssist technology. We're very excited about this technology and what it offers. My background, very quickly, I spent about 15 years in surgical pathology as a PA and 4 years as a histotech, and then a little bit of research time in pathology and immunology as well. So let me dig in here. Start with spatial gene expression with Visium. So what we're looking at here, this is an H&E stained image of breast tissue, human breast tissue. This is significant for DCIS. And our Visium spatial transcriptomic solution, what this really allows you to do is to create a sequenceable library from tissue sections like this. It can be FFPE sections, it can be cryo sections, and then to get the whole transcriptome profile from these tissue sections across the entire sample itself. So what you see on the right, this is a sequencing readout that's overlaid with the tissue section. And this is based on hundreds up to thousands of RNA markers here. And this is what really lets you start interrogating the sample to see where your immune cells are, where cancer cells are located, what might be happening in stroma that's adjacent to areas of tumor in a duct. Perhaps. So this is really the power of Visium. And again, this looks at the entire tissue section. It's not a region of interest based approach. It's designed to really bring out an unbiased level of data. So here we're looking at H&E. This can also be accomplished with immunofluorescence. In terms of the options here, many customers will utilize maybe 2 antibodies and DAPI. However, our software is capable of detecting 6 colors, so 5 antibodies can be used in conjunction with DAPI as well. In addition to clusters, you can, of course, utilize our software, which is free, right? There are no licenses required for our software. And you can interrogate the tissue in this way as well. You can enter specific markers. As an example, on the left, we're looking at HER2. The expression in that sample, and you can see how that correlates with a prior image of breast cancer in areas that have DCIS located within them. Similarly, you can look at other markers as well, find where these genes are being expressed and the level of expression as well. And it's this type of discovery that helps advance efforts in studying different treatment options out there. Some uses in the field. I have a few here to show you. This one is out of Satoshi Nagasawa's lab at the University of Tokyo, and they spatially profiled DCIS in human breast tissue. This revealed the existence of heterogeneous cell populations, which provided predictive markers for classifying the disease. and also improving efforts in treatment research, as I mentioned earlier. Carberg and Pierce out of Stockholm, Sweden, they looked at synovial biopsies to look at conditions of rheumatoid arthritis and also spondyloarthritis. And by looking at these biopsies, they were able to find novel gene expression characteristics of both these inflammatory conditions. Faulkner, Corbett, and Pierce out of University of Oxford, they spatially profiled the development of the human gut, of human intestinal tissue. They utilized full thickness sections to really appreciate everything in the wall from mucosa to serosa. And they were able to identify many critical insights into the development. And that includes epithelial crypt villus formation, differentiation of mesenchyme, expansion of vascular systems, colonization by immune cells, and also development of those propria, those muscle layers within the bowel wall as well. So the way the technology works, this is actually what we refer to as our V1 slide. This is the original Visium slide, and it shares the same technology as some of our updated ones. So I'm going to touch on this briefly. So many of you are familiar with this technology already, and what it utilizes on the V1 slide is a direct tissue mount process. And this leverages 4 capture areas that are on the surface of the slide. Each are 6.5 by 6.5 millimeters. And each contain about 5,000 barcoded spots. Each spot has a diameter of 55 microns, so that's the resolution of the slide. In terms of any technology out there for spatial transcriptomics that allows you to take a picture of that section and move forward with library construction, this is the highest resolution that you can achieve right now. Downstream of that, there's a poly-A capture for fresh frozen tissues that's completed with these probes. And there are millions of these probes in each capture area, in each one of these barcoded spots. And what's unique is the spatial barcode to it. That maps back to that specific spot in the capture area where transcripts are captured or probes are captured in the case of FFPE tissue. The 2 options, there's fresh frozen tissue, that's our species agnostic solution. In addition to mouse and human, multiple other species you can work with that benefits of really strategically embedding your tissue in an OCT medium, especially if there's not much experience there. FFPE tissues, this has really opened the doors to working with archive specimens. So clinical specimens, research specimens from human tissues particularly, but definitely targets the full transcriptome both for human samples as well as mouse samples. Workflow is pretty straightforward and it's unique in the way that it's integrated in labs. It essentially starts with scoring down blocks to the size to fit in these 6.5 by 6.5 capture areas. Downstream of that, you have a staining option of either H&E stain or immunofluorescent stain. Then you grab a high quality image there. Downstream of that, you complete some barcoding, some bench work, construct your library, and then submit that for sequencing. Once that sequencing readout is obtained, our software will align that with the image automatically. There's also a manual alignment process. But essentially, you end up with an image at that point that is very similar to what I showed you earlier with the breast cancer specimen. So before I get into cytosys, I just want to cover a key part of what's happening on the Visium slide. We're looking at the permeabilization process here. This top row that you see with fresh frozen tissue, this describes how fresh frozen tissue is processed on that direct mount slide. The tissue is placed on top. We have some workflow steps that we introduce different reagents. We permeabilize the tissue and transcripts are captured directly on the surface of the slide through poly-A capture. With FFPE tissues, it's a little bit different. We introduce probes. So those probes are hybridized to those mRNA targets. We permeabilize the tissue, and then those probes are what diffuse down onto the surface of the slide. So again, this is the V1 slide where tissue is placed directly onto the Visium slide surface. All right, so here's cytosys, and this is the technology we're very excited about. There's producing great data, great results, and utilizing a variety of specimens. So this allows researchers to analyze samples mounted on routine histology slides. So that means no more scoring of blocks are necessary. You don't have to trim down blocks and deal with that type of challenge that it can present, of course. We're also excited to talk about some upcoming updates to this platform, which I'll get into. One we're particularly interested in covering today is the multi-omics offering. So this allows users to appreciate morphology through H&E staining or immunofluorescence, RNA detection, and then protein detection as well, all on the same tissue section. So I'll have some interesting data that we can look at as well. Right, so how does CytAssist work? This all starts actually with the new technology of the V2 slide. There's some changes to this, some features, and I'm sure some of you noticed right away. There are 2 capture areas here instead of 4. But instead of just 6.5 by 6.5, you also have an option of 11 millimeters by 11 millimeters. This definitely will provide options for more discovery, or you can put more samples in there as well. It doesn't have to be 1 small sample or 2. You can maximize that and definitely utilize the whole real estate on that slide. What else is unique about this slide is there's more options for human tissues, more sensitivity. The probes, you have up to 3 times the amount of probes per mRNA target when you're working with human tissues. So you have improved sensitivity. And for those who are familiar with the DV200 score, that's an RNA quality score. With Visium enabled through CytAssist, that score can be 30% or greater instead of 50% or greater. So it's tissues that may be impacted perhaps by a fixative used alongside of formalin, or something that may have been decaled slightly or affected by cold ischemic time, those are now opened up. You open up access to more specimens with a little bit lower RNA quality to them. The way it works with the workflow is that you have 2 tissues that are placed on standard slides. These can be superfrost, they can be uncharged, any type of standard histology slide. And then you have your Visium slide. These are placed into the instrument, and the CytAssist instrument enables transfer of probes from this tissue on the standard slides onto the surface of the CytAssist Visium slide, which I'll refer to as the V2, the version 2. That's what we refer to it now. Output is your Visium slide with analytes on top. And then the CytAssist also captures 2 images as well, as soon as the run is complete, actually upon closing the lid. And I'll touch on this part of it as well to give you an inside look. So after your slides are placed into the instrument, they're placed on the top surface, which is underneath the lid. Visium slide is placed on the bottom. We introduce some permeabilization enzyme. Close the lid. That's where a couple of images are captured, and 30 minutes later, those analytes are transferred from the tissue onto the surface of the Visium slide. Then you have your outputs, as I mentioned before. In terms of the workflow, this is very unique. There's a lot of opportunity to expand the tissues you're working with here. This front end portion here, when you're working with this slide here, this can be cut fresh at a site, at a lab. They can section an FFP block, put that section on the slide, move forward with it. Additionally, if collaborators are working with a location that has CytAssist, they can cut sections onto a slide and send those slides in. Since you're not working with a slide with a delicate surface to it, you can now put it on a plain slide and send them in, whether it's across the city or it can be from a other end of the country, from the East Coast in this case. So it really opens up the options there. The data we are seeing off of this is remarkable in terms of replicates, consistency, and reproducibility. And I'll touch on some of those topics shortly. In terms of what you can gather, instead of not just freshly cut blocks in-house or sections that are placed on the slides and CytAssist also has a workflow which enables the use of archive slides. So slides which have been stored up to 1 year that are FFPE, H&E stain, and cover slip. And this is with that cover slip medium that's actually intended to hold slides for multiple years. Our protocol effectively allows you to de-cover slip those, remove the adhesive, and put that right into the workflow. The H&E and IF staining, of course, are options to appreciate your morphology in the tissue. And then you can choose the best section as well. So with the prior method, putting sections onto the Visium slide, you'd have to revisit the block after you previewed a section and take a deeper section. With this new method, the tissue you preview on the plain slide is the same tissue you can move forward with and obtain your whole transcriptome profile from that exact same section. The slide itself, as I mentioned, improvements in the RNA quality you're working with, sensitivity of human tissues, and then also that flexible area that you can work with on the slide surface. So the data that we're seeing coming off of CytAssist, both for immunofluorescent stained or IF stained tissues as well as H&E, is quite remarkable. We are consistently hearing positive feedback in many ways. It's also, there's reproducibility that's introduced. And that's at that point where we introduce that permeabilization enzyme. It's a very critical part of the workflow to be able to reproduce that whenever possible. And we're able to do that with CytAssist. The demarcations, as you can see in the tissue, are quite a bit stronger. We have more minute structures, which we can appreciate, minute anatomy in these tissue sections. More germinal centers are being identified in lymphoid tissues, cryptic structures in this tonsil, for example, and patterns which are consistent with the histology you'd expect, maybe for the mouse liver or the, I think it was a mouse testes as well. There's this reticulated pattern that you see that it almost looks like multicolored histology. It captures so tightly the transcripts that are coming across or dropping down from that permeabilized tissue. We also see improvements in the data. What you see on the top row is a stained ovarian cancer section, and down below too, it's a little bit more intense staining here, but that doesn't impact the assay at all. What we see on the top with direct placement, you see clusters, you see good separation. Our direct placement is still a good option to complete discovery on your sample. However, we're certainly seeing an improvement in our data. As you can see, there's more separation with these clusters, a little bit cleaner demarcation in the clusters for the CytAssist enabled workflow. Similarly, we have some other tissues we looked at here where we compare this direct capture method. So again, this is the blocks that were trimmed down and sections were placed directly on the slide. Still provides great data. However, when we test the same or deeper sections from that same block on the CytAssist enabled workflow, we do see improvements. We see more these minute structures coming out. Again, in this case, we're looking at germinal centers. Here, more separation in the ovarian tissue, and then some more lymphoid tissue here. Lymph nodes, you see more germinal centers, especially small ones to the periphery over here. Right. I want to revisit a couple of the value points here and some of the changes and the advantages of working with some other parts of the V2 approach. One is that larger capture area. What you see in the smaller capture area is about a 5 millimeter punch sample from a human breast cancer specimen. The larger area, the 11 by 11, shows you really how much more discovery can be completed on that specimen. You can really capture more from a single specimen and also put more tissues in that sample as well. We have some advancements in that regard, which I'm going to cover shortly as well. Now, in terms of archive slides, this is a sample of lymph nodes, which we put through the Visium workflow enabled by CytAssist. We had decover slipped a slide that had been sitting at room temperature with a cover slip on there for 5 months. Again, slides are validated to 1 year. It doesn't mean over 1 year won't work. We just haven't validated that yet. We are looking at slides well past that point also. In this case, you can see we looked at markers RGS13 for B cells within germinal centers, and then CD19 for other B cells, a little more diffusely located through the specimen. And we have good spatiality in both sections, despite that time under cover slip, despite that time under that harsher medium. Our probes are still able to effectively access the mRNA once we decrosslink and depair, or once we decrosslink that tissue. Little more data where we focus on immune cells here. Dendritic helper Ts and then macrophages we were able to identify within these sections that have been on archive slides. So we have some exciting updates as well with CytAssist. We recognize there's a need for some other sample types that customers are interested in. Here what we have is an example of a tissue microarray. So this has been of interest for quite some time. Customers have wanted to work with TMAs. It definitely can be a challenging specimen to work with. It takes time to build. Sectioning can be a little bit tough. These sections tend to curl underneath, and the idea is to create as little disruption as possible on these type of sections, and Site Assist allows that. It allows you to cut a section, float it, pick it up on a standard slide. What we see here is a 5x5 or 25 total cores TMA, and we separated these out. We used brain tissue and kidney tissue in this sample, and we were able to restrict this analysis and separate the data with brain and kidney, but we could also take it a step further there, where we could look at the specific cell types within each tissue type as well. So as you can see, we separated some of the tubules, proximal tubule, the nephrons as well within the cortex, and then in the brain, we separated out the cells present there as well. And with our software, you can restrict that analysis per core or to certain cores or to certain tissues that are part of that. All it involves is just tracking the cores that you have in there. Another exciting update is the option to work with fresh frozen tissue. So this time, if you have mouse tissue or human tissue that's frozen, you can move forward with the Site Assist enabled workflow. The way we do that is we complete an on-slide fixation step. Following that, you can utilize the FFPE kit and reagents for that tissue that was previously frozen. Again, provided that it's mouse and human at this time. So those probes will hybridize those mRNA targets. Some unique aspects about this, for anyone who has worked with our prior FFPE option fresh frozen option on the direct placement, there's no tissue optimization step required any longer when you're utilizing this workflow. It's a standard permeabilization time. The RIN score also used to be a requirement of 7 or greater to move forward. That's changed now. You can work with fresh frozen tissues that have a RIN score of 4 or greater. So tissues that have been sitting in a sub 80, for example, for quite some time, I've seen them drop down to 6 or 5. Customers want to move forward, they can't, but now this enables you to do that. You can definitely leverage archive FF or fresh frozen tissues, and then also the benefits of cryosectioning, as I mentioned earlier, to strategically embed tissues or just manipulate them in a way so you can really maximize the area on your slide, and then also then leverage the 11 by 11 now with fresh frozen as well. In addition to that, we do see improved data where we see on top here, in terms of space, we're looking at the Site Assist enabled breast cancer specimen. And then on the bottom, this was a direct application from poly A alone. On top, we switch over to the probe base. You can see the assay with Site Assist. This does provide better mapping of spatial information, as you can see by the sharper demarcations and spatial patterns there. On the bottom, when compared to the bottom, you can also see that the annotated regions here on the left from the pathologist, they do align more closely with what we see coming off the Site Assist enabled workflow. So altogether, with the data we're seeing, as I mentioned, we're very happy to see the level of detail we can capture in this tissue. We continue to get great feedback. Well, I'm showing you here our tested tissues for FFPE specimens. And this is, of course, going to grow once we start adding images of fresh frozen tissues coming off of this platform as well. This isn't an exhaustive list, right? There are multiple tissue types that are being or tissue sources from human or mouse that are being brought into this workflow that aren't represented here, but it can definitely be put through. Another update that we do have is that PFA fixed tissue will be enabled soon. We're working on that. So fixed frozen tissue that is PFA fix. We are working on an assay to facilitate that too. Right. The protein. We're very excited to present this. As I mentioned earlier, this is our multi-omics offering. We have morphology that can be appreciated with H&E, with IF, and then we have RNA detection and protein detection on the same tissue section. We will launch about late Q2, about spring of this year, with a 35-plex antibody panel for human immune cell profiling, along with the ability to spike in protein targets that are not in the panel. And we're just on the verge of providing that list, a little preview that will target macrophages, dendritic cells, B cells, T cells, and many other immune cells out there. So we'll be sharing that pretty shortly. An example of how we've utilized this, here we extensively characterize this NGS-based protein readout and human immune cell panel across a wide variety of samples. We cultured these two cell types here, you see on the left. And what we did was we compared where we were seeing CD3E against PAX5. You can see we were able to effectively differentiate those between these two tissue samples here. These are cell pellets that we created from cultured cells. And you can see we have good separation there by applying our panel. Additionally, we looked at some utilizing our TMA process. We created a small TMA of different tissue types here of heart, spleen, tonsil, and breast cancer tissue. And through our panels, you can see we're able to identify specific expression of PAX5 in the tonsil, CD68 in the spleen, PDL1 in the heart, and PAN-CK in the breast cancer punch. On the larger sections that you see on the right here, you can see that CD3E is located primarily external to germinal centers, whereas PAX5 is seen both in germinal centers as well as in other parts of the tissue, as would be expected in this particular case. And we're able to accomplish this through the use of this immune cell profiling panel. Right, so to wrap up on CytAssist, I do want to touch just briefly on some of the advancements here and updates. Again, there will be upcoming guidance on TMAs. And we also have that fresh frozen capability, which means no more tissue optimization, RIN score of 4 or greater, and then leveraging that larger capture area for your samples. We'll also be supporting PFA, fixed frozen tissue, in the future. So that's in the works right now. The multi-omics offering, as I mentioned, H&E and IF for your morphology, RNA detection, and protein. And again, that will be on the same tissue section. And then then the larger capture area and any type of guidance that we can provide in terms of specimen preparation or experimental design, that will always be available, as will the support for 10X. It's always a point of pride for us. Any place we can help, we have teams and FAS built around these technologies for spatial, so we'd be happy to support you however we can there. So with that concludes what I have, but I think we may have some Q&A.
Yeah, how does that work? In terms of the Q&A, we just open up the floor. Would they actually you'll be from your computer, perhaps?
Yeah, let me take a look.
Yeah, so now if anyone in the audience have a question, or maybe here in the room on site, anyone have a question, please feel free to ask John.
Okay. All right. I see a question there. Where would the audio? I see Joel. Here we go. Is there data available to? Yeah. So the question is there data available to play? I think to see what's what we're capable of, some of the data we produce. We have multiple data sets that are available, and we can share a link on that. So multiple tissue types, data, and also a link to our software, the Loop browser, where you can download the latest version of our software. You can explore different data sets and really get an idea of how you can interrogate tissue specimens and samples. Depending on what you're working on, we may have data sets for that particular tissue. So yeah, we definitely have quite a few options for you, and we can share that information after our call today.
Hi.
I was
just trying to see that we like that sound come, but I don't think that I can hear the sound.
Yeah, I don't think I have audio. If it's possible to enter the question in the chat. See, I don't think. Okay, multiplex 2 samples in one capture area. If the question is, can you multiplex 2 samples in one capture area, 6.5? So two tissue samples can be placed into a capture area. We'd recommend, if I'm understanding the question correctly, two samples can be placed into a capture area. However, the optimal way to approach that is through embedding. So if you can embed that in a way where you can maximize the number of samples in one section rather than putting sections into one capture area, that's the optimal way to proceed.
Mm.
When would the PFA? Okay, question is, when would the PFA fixed fresh tissue protocol be available? We don't have a firm timeline yet on that. However, as soon as that comes out, we'll let you know. We know it is next in line, so we currently launched the fresh frozen. So fixed frozen is next up. So that's where our efforts are geared at this time. As soon as we know, we can send that information out. Right. And what is the maximal thickness of the tissue? Can you do 3D rendering? Right. So 3D rendering, that's not something we can do with our software at this time. And that's because we don't have a Z-stack component here. It's a completely two-dimensional image. I can definitely share that feedback with our team to let them know that people are interested in that. In terms of the maximal thickness of the tissue, that depends on the tissue you're working with. For fresh frozen tissues, we recommend starting at 10 microns in thickness. For FFP tissues, it's 5 microns. There's some wiggle room. The direct mount process, you can, it's 4 to 8 microns. And then with the site assist method, anywhere between 3 to 10 microns. Question about the, okay, now I see a question that says, I have a question about the tissue type. Have you ever used mouse lung tissue slices with Visium site assist? I have not worked with someone personally who has done that yet, but I know someone who is planning this. And what I can say is we've had multiple customers use mouse lung on the standard direct placement on V1, and they've been very happy with their data. Data's look really nice. The sections look really nice, and I only anticipate they're going to look even better coming off the site assist instrument. Is the RNA, next question is the RNA liberated from fixed samples by enzyme treatment? So in fixed samples, what we do, we hybridize the probes. From that point on, after we've hybridized them, we permeabilize the tissue, and the probes are then captured on the surface of the slide. From that point on, what we do is a DNA extension to capture that information and create our library from there. So the RNA is not liberated. What we do is hybridize probes to that RNA and then create a library from those probes that are captured from the tissue during permeabilization.
So John, regarding the probes, so these are the sequences of probes. This is so comprehensive, so it covers all the transcriptome.
Yeah, these for the probes, they're designed to cover the most conserved regions of the protein coding mRNA for whole transcriptome for human. Yeah, that's how they're designed. A little more on the probes. They're about hybridization region total is 50. We don't have the specific measurements of the left-hand right side because they're a pair, but yeah, they're designed to target the most conserved regions of protein coding. Yeah.
Looks like that leads into the next question here.
Oh yeah, what's the next one? I wasn't clear if we could profile the entire transcriptome or select set of transcripts based on probes, both for human and mouse. Yeah, so it's whole transcriptome, and the targets are the target mRNA are protein coding portions. Yeah.
They are select subset that's not covered, right? Like VDJ, fragment, those kind of gene, and mitochondrial gene are not covered.
Yeah, I don't think mitochondrial genes, because the size, yeah, it would depend on, yeah, I think it would depend on the size that they're after. If we had that information, then we could probably speak more to that. We do have some customizable options that have just come out. So initially that was for fresh frozen tissues, but we have a tech note now for FFPE tissues. And then with that, we should be able to, in addition to, yeah, bacterial and also viral transcripts, we should be able to target.
Mhm.
Great question.
Any other questions?
Question about the protein detection. So on your DNA or
Yeah, the plan for that is DNA barcoded antibodies.
Okay.
That's what we'll utilize, and then libraries will be constructed in parallel. Yeah.
John, a quick question about the comparison data that you have between the direct placement and the FAP, the cytosist
Right.
data. For the direct placement, is it a V2 probe or is it a V1 probe?
Or for the direct placements, yeah. So I'll answer this. So the probes are being used for human on V1 are different
Yeah.
from the ones that are being used on V2. The
Mhm.
ones on V2, you can have up to 3 probe pairs that bind to mRNA targets, and it's just one set of or one probe pair per mRNA target for the direct mount method. For mouse, they remain the same at this time.
I see, right. But then because I'm more talking about specifically for the data that you showed earlier in one of the slides. So there's a comparison.
Right.
That is actually a direct comparison between V1 and the cytosist, or the replacement with V2 and cytosist with V2.
Okay. I'm not sure I'm understanding the question. Is the
Because she doesn't, we had a comparison there between BD1 and
Right.
sustainable, but we're using different transcript sets, it seems like, right?
Yeah, so because the question I guess is to understand how good the cytosist is helping better than the replacement. So if the comparison is all both using the V2 probe, then it's fair comparison, right? So that's why I'm asking in that data that you show. I'm not sure if that's actually using the V1 probe or V2 probe.
Oh, got it. So that data, the cytosist image is using the V2 probe.
Right.
Yeah.
But the replacement is using
The V1 probe.
the V1 probe.
Yeah.
Okay.
So in the V2s, that's what's giving us better sensitivity as well,
I see.
and for that
Mhm.
human sample.
Okay.
That's a
Yeah.
great question, but
Yeah.
and we have folks that have applied this already, some of my other customers, and they have done the comparison where they've taken those serial sections
Yeah.
and gone both pathways to really be
Yeah.
stringent with their comparison, just like you're asking.
Yes.
And, because they already know what they should be expecting, and they are seeing that, yes, there are different probe sets, but when they actually compare the serial sections on both pathways, they do see better clustering, and they do see more sensitivity.
With cytosist. Yeah. Great.
That is a great-
Yeah, great.
Sorry, I have a question. Can we use the V2 probe on the direct placement?
Yeah, we actually designed that very specifically for the cytosist workflow. There's some changes. You have some different temperatures that are utilized during the permeabilization process. And it's for a shorter period of time as well. So we designed those based on the workflow specific to cytosist. We don't know necessarily how it would work just because we haven't tested it internally on the manual method. We wouldn't advise that, all right, just because we haven't worked through that in-house, we haven't validated it, we haven't tested it. So it's something we'd recommend sticking to the cytosist enabled workflow when you're using those, the V2 kit.
So there's another new question on the chat, the Q&A.
All right, the question here. So the slice thickness, so we currently work on mouse pressure cut lungs. The slice thickness 300 meters. We're tracking how fast V2 cells decreasing during these. Do you think this? Well, 300 microns. I mean, I'll start there. Really, that's we haven't tested anything close to 300 microns, so that would be, I wouldn't recommend that for our workflow. It would depend, I guess, on if 300 microns is required, if it's not required, but what I can tell you is 300 would be on the thick end for sure. That's not anything that we've tested. I've heard of some customers moving forward with 20 microns, and you can actually get up to 20 microns for the fresh frozen assay, which is coming out, or the fresh frozen tissues, which are enabled. 300, I would say, wouldn't be a good idea to move forward with. Yeah, we don't have anything to support that at this time.
For their study, they could use some serial sections and be able to say whether
Yeah.
the cells track into that region, right? By look.
That's a thought. Yeah, yeah, yeah. If you can serial sections across maybe 2 slides worth, if you were to use fresh frozen perhaps, yeah, that's something we could definitely talk about in terms of planning and maybe coming up with an idea on how you want to proceed.
John, I have a general, maybe stupid question. So you show one slide, you have the RNA, DNA, and then you have the protein.
Right.
So how close are those two correlated? For example, on the RNA, DNA level, say you have a mutation you found, and on the protein level, do you found the same number of protein species correlate all what you found on RNA, DNA, or it's different?
Yeah, it's a little early to say for sure on that. It's going to be launching in spring, so we'll have more data there. Yeah, it's a little early right now to tell you on that specific sample. I can ask to see if we have some more information there. Yeah,
Thanks.
yeah. We should soon though. Yeah.
Thanks.
There was a publication a few weeks ago now where we looked at carcinoma institute PCS. And we're looking at glial cells, there really good paper where they looked at single cells, Visium and Xenium. And I think some of the work there shows that there's a really nice correlation between the gene expression and the protein. I can do that.
Oh, that will be great, thank you.
But it was some of the general markers of the cell structures. I think some of the protein markers, gene expression ones, which are very plastic, might not match up in time because even within the cell, they're all temporal, right? But some of the main cytoskeleton show very clearly like the overlay.
Okay, so we will break for lunch and hope you guys can join us back around 12:40 or 12:45. Let's do 12:45, yeah, for 1 hour break.
Sounds good.
Yep, thank you.
There we go.
You have 20,000?
So I don't. Gosh, that's magical. How do you? How do you think? You. You a program, are you a computer?
My.
Under analysis, like user. Okay. You want
You were,
to-
you guys are over there in East Bay, right?
No work area.
No,
Yeah.
there.
I know,
That's where,
yeah.
what is the, new
Here,
sequencing place? Ultimately, no.
Yes, yes, exactly.
We were in a.
Thank you.
Everybody is porting 10x for sequencing, Ultima, Element.
Well, a lot of folks actually went to Element.
Element was actually in our facility. When they were in Stanford, they had a little plug in our lab.
A scientist, and her building was mostly owned by her company, but they rent out some of the lab space.
I was just like that too.
And she recalls that one of the labs, that tiny little startup that rented space, was a company that had a bunch of spiders. So I used to freak her out. She'd be like, why does this company, like, imagine the lab down the hall was just full of giant orb weaver spiders.
Oh,
They are breeding spider?
No, they were both, they're both,
Scary, but
it's they were trying to understand the protein molecules
Oh my
for
God.
strands. that they
Yeah.
did. And they did
Yeah.
figure it out, so they don't use spiders anymore.
Oh my God.
But they did it so they could sequence and find out the protein structure.
Yeah.
Capitalize on those structures.
Yeah.
Yeah, but at that time, 12 years ago, they had live orb weavers, the big ones everywhere.
That's
Those like
crazy.
the big giant ones.
That's
I
crazy.
hope they didn't escape one of the.
Actually, those are not venomous or poison
Poison.
at all, right? When you go to the Academy of Sciences, they have them out just
Right.
doing their thing, right?
But I think all the movies make spiders so scary and kids.
She just said you guys are not on mute. So if you guys are just having conversation.
I can, yeah, let me start to share and then I'll just, maybe slowly introduce in, yeah. Apology for little bit late. Still have a few people now not in the room, so we'll be probably just waiting for a couple minutes. Okay, everybody. We are back for the second talk of today's webinar series. So my name is Jasmine Chen, and I'm the Director of Genomics at the A Biosciences. And today I would like to introduce you our A Bioscience spatial capabilities. In the A Bioscience US Genomic Core Lab, we are providing, oh, sorry. Sorry, that's not showing. We are providing the single cell genomic service as well as spatial transcriptomics. Sorry that I'm just trying to start the projector in our room here. Can you guys see now?
Yeah.
Okay. Seems that is. Yeah. What we do here, as illustrated here, that we receive different type of sample from clinical sample from the animal model, sometimes xenograft, and we receive some of this tissue through the logistic, and we then can either dissociate the tissue into single cell suspension, or we sometimes receive fresh or cryopreserved sample that are directly from our customer. And these sample can then be further treated in-house through the compound treatment or antibody staining. We can also FEXOR or do the magnetic bead salt in order to enrich certain cell type. We can also do the multiplexing. These samples then be proceeded into the single cell genomic work, and we can actually handle, as you see here, multiple different applications, including the single cell RNA-seq, single cell DNA-seq, attack-seq, and immune repertoire through the site-seq experiment. And we can also provide the CRISPR screening through the single cell and the multi-omic approach that can combine the attack and nuclei. We also have done quite a bit of the multimodality that can assess RNA, TCR, BCR, and the protein from the same cells. The newest application that we have developed and validated in-house is the single cell fixed RNA-seq, which we will talk about in the near future together with 10x Genomics. So this is actually using the FFPE tissue where we isolate the cells and nuclei, as illustrated at the bottom here. And for the spatial transcriptomic, our laboratory are capable of working on both frozen tissue and FFPE tissue. And from here, we can isolate nuclear cells from frozen tissue, and this we can do the single nuclear RNA-seq. And we can also perform the 10x fresh frozen spatial transcriptomic. And by combining these two, we can utilize the single nuclear data to deconvolute the fresh frozen spatial spot. And actually, Nassim will talk about some of the bioinformatic work that we done in-house, and she will talk about it in the second, in the following talk. From the FFPE tissue, as I said, we have this capability of isolating cell and the nuclei from the FFPE tissue. And now we have the new is the cytosys FFPE VCN, which is what I'm going to talk about today in the talk. I will not talk into too much detail on Nassim. We'll talk more in this, but just to let you know that in A-Bioscience, we are also offering the single cell bioinformatic analysis service. And we also have the Omni browser and Omni database. And our bioinformatic team are very strong. They are actually capable of providing customized analysis. And we have a very solid pipeline to help the customer to analyze their data in all the genomics data. So just briefly, I think earlier John has talked about a lot of application about how the VCN data can help to understand. And so I'm just gonna quickly tell you two of this paper that really illustrate the power of the VCN. The first paper is this paper from the Jacqueline Landberg and the Paul Cavari. And they are analyzing the human cutaneous squamous cell carcinoma. They profile these through the single cell analysis as well as VCN. And so from the single cell data, they can find this tumor-specific keratinocyte. As you can see from this figure, this cluster 10 and cluster 7 here from each of the patient have a very specific keratinocyte score. And then they use this to look for the signal at the VCN. And then they can actually find that these tumor-specific keratinocyte high cluster spot are located at the tumor leading edge. And this actually helps to understand about how the tumor are developed and are different from the normal. And furthermore, this paper in that I'm showing here, they are actually using the VCN data to provide a high confidence cell-cell interaction. So this is a paper done by a large group of collaboration, including Sarah Tachman, Margarita Turco, and the Rosa Bento Tolman. And they actually analyzing the human endometrium in a very extensive way. And they have actually traced multiple time points through the menstrual cycle. And they found out many different types of cell through this single cell analysis. But from the VCN data, what they can do is actually use the spatial data to identify the cell location, and then use the cell phone DB version 3. They then look at the combination of cell-cell interaction. For example, shown here on the top, they first of all do the differential expression interaction between ligand and the receptor. And then they look for cells that coexist in the microenvironment, for example, that are located in the same spot. And so this actually provide the confidence of the cell-cell interaction. And then they look for the protein ligand and receptor interaction through this cell phone DB. And then they can actually find the interaction between this protein receptor and ligand. And so this is actually a very nice illustration of how the spatial information can provide us more deeper analysis of the different tissue and cell type interactions. So in ABio, we have been adapting the 10x VCN for quite some time now. Starting in 2021, our team had already started to work on the fresh frozen tissue. This is established at the Q1 2021. And we have done various different type of tissue in our company, helping the customer to understand their tissue type, including in the human, liver, lung tumor, and biliary tumor, and gastric tumor. In the mouse, we have also looked at various different tissue, including the normal liver, normal heart, normal lung, and the colorectal cancer, and the lymph node. And in the summertime 2021, we quickly adapt the FFPE VCN. And after that, we have done much more extensively using the FFPE tissue. And as you can see that we have looked at many different type of tissue in the human and the mouse. We just started to work on our cytosis. This is our new capability. And so far we have just worked on 2 different tissue, the gastric tumor and the liver cancer. But we believe that we will have much more to come. And we are very proud to be the VCN in spatial solution service provider for 10x Genomics. So I know that John has probably talked about a lot on cover of some of these, but I wanted to talk about it just for those people who did not join in the morning. But again, to emphasize the differences between the VCN fresh frozen V1 and the cytosis. So illustrated here, just to show that the VCN is really utilized in the same slide here. There's this capture area where we have 5,000 spot for the 6.5 by 6.5 millimeter capture area, or the larger one, 14,000 spot for the 11 millimeter by 11 millimeter capture area. Each spot is a 55 micron diameter spot where they have the spatial barcode, UMI, and the poly-DT primer that are decorated on the spot. And for the VCN fresh frozen, it's utilizing the poly-A that are released from the tissue. And then they hybridize to the poly-DT primer. And then we utilize the template switch oligo priming to generate the RNA library for the whole transcriptome. With the VCN FFPE for both V1 and the cytosis enabled chemistry, they are using the what they call the split probe chemistry. There's a left-hand side probe and the right-hand side probe that are ligated and hybridized to the target. I'm sorry, it's hybridized to the target first and then ligated. And then so these enable the FFPE tissue to be detected in a whole transcriptome manner. And this actually also because of the pro-hybridization. So you don't have to worry about FFPE being the more degraded sample type. And so the data has been pretty powerful to enable the archived FFPE tissue to be studied with this VCN technology. And so the notable difference between VCN V1 and the cytosines is illustrated here. For the V1, there's only 1 probe per genes. At the cytosines, there are 3 probe per genes. For the VCN V1, it's a direct placement on the VCN slide. But from cytosines, we are utilizing the section from regular slide. And this can either be freshly cut or archived slides. In the V1, there are 4 capture area per slide. But for the cytosines, there's only 2 capture area per slide. There are more choice for the cytosines in terms of the size of the tissue. We have the 6.5 by 6.5 millimeter or the 11 millimeter by 11 millimeter, which have much more spot, almost 3 times more of the spot than the 6.5 by 6.5. The DV200 recommendation is lower now with the cytosines. Now we can actually apply tissue that has lower DV200 down to 30. And then in the V1, the DV200 recommended is 50. So now I'm going to talk about our in-house validation of VCN by cytosines at A Biosciences. Here I'm showing the 2 tissue that we have in-house that we use to do the validation. Both of them are gastric tumor. On the left, it's an invasive adenocarcinoma. It's a smaller tissue. And on the right, it's a larger tissue that you can see. It almost expands the entire slide and it's much larger. The age of the block are quite different. As you can see, on the left is 2 years. On the right is 11 years. We also have a section that we have cut and then we just archive. We already H&E and we just keep it on the side. And after 18 months, since the cytosines was available, we decided to use those slides that has been cut 18 months ago to validate the cytosines workflow. And people might wonder about the DV200 from the fresh section versus the decovered sleep archive tissue. So we have this data which we have obtained from the freshly sectioned tissue. Here's the DV200 of various type of tissue, including the human gastric, a few different mouse column. Then we also have those slides that we already H&E stand and was mounted, so the keep under the room temperature, just as John described earlier. In here, we have tested the age of slides is 14 months to 7 months, and we look at the DV200. They are still very similar compared to the freshly sectioned tissue. But with the mouse column, you can see that they are slightly degraded, but still pretty good enough to do the analysis. Another question that we have often hear from the customer, how big do we need in terms of the material for the RNA score? So we here provide all the information, including the size of the tissue. This is just the surface area. All of them are 5 micron section. And so some of them are larger, some of them are smaller. But we also have tested, just as 10X suggested, if we just do a small scrape of the tissue, as opposed to a curl, the entire curl of the section, which as an example here, you can see that we have a larger tissue, 48 millimeter curl, as opposed to the same tissue that we just take 4. So it's a 2x2 section that we take out for DV200 measurement. And you can see that they are still very close to each other in terms of DV200 score. So this is actually pretty good. You can feel satisfied and feel more comfortable of using the same section that's partially used for cytosis and partially used for determining your tissue quality using the DV200. So yeah, so here just again to illustrate the differences between the two platforms. On the top is the VCN V1. Quickly, as we already said, the tissue have to be directly placed on the VCN slides in each of the capture areas. So we can put 4 tissue here and then go through the workflow, including H&E staining or immunofluorescence staining, imaging, and then go through the hybridization, ligation, barcoding, and library construction. For the cytosis workflow, we can either take it from the FFPE with freshly sectioned them on regular slide, or we have what we have in our example here, a pre-mounted tissue slide that has been stained with H&E. These 2 slides are then placed onto the cytosis machine after the hybridization, pro-hybridization, and ligation. And then the VCN, cytosis VCN slide will be placed on the bottom of the cytosis machine. And then the machine would enable the RNA transfer, the ligated probe transfer from the regular slide to the VCN slide as shown here. And then we can proceed with the library preparation through the workflow illustrated in the 10X protocol. And this is a more detailed workflow that are shown here. Again, we would image the pre-mounted tissue slide. We would then decover slip and then do the decross linking. Then we apply the probe to perform the pro-hybridization and ligation. Then we use the cytosis to transfer the ligated probe and then also do the permeabilization. And then the probe extension is done on the slide, and then we will do the preamp in the tube and then the library construction. Here's to show you the QC data that we have after the library construction. We have seen a single peak of the library after the workflow from these 2 tissue. And I'm going to quickly show you the results where we compare the V1 and the cytosis. In the V1 version, we also have done with these 2 tissue that are shown, the data on the left. This was in the blue. And then the cytosis data on the right, that's highlighted with green. And they are very similar tissue, except for V1, it's actually more exhaustive because we have cut through the section. So the tissue are smaller in the V1, but for the cytosis, it's on the opposite, it's larger because it's an earlier tissue where we just H&E and use it for pathology annotation. And now we are actually using the same tissue where we have pathology annotation to be applied on the cytosis. And as you can see, immediately you can appreciate the power of the cytosis. Looking at the genes, median gene per spot, for the gastric 251, we only detected 3,000 gene per spot. But with the cytosis, we obtained about almost 7,000 gene per spot. For the AA2, because the DV200 is lower, so we actually only obtained 400 genes per spot. But compared to the cytosis, it's really increased to 3,000 genes per spot. And we also have all the cell space ranger that we can actually just to share that these are the data, but I would not go into detail because this is not really our specialty. Nassim will talk about it in a later talk. But just to show you that, these are the results from the V1 of the 2 different tissue. And as you can see that these 400 genes per spot are not as, the cluster is sort of, hard to distinguish from each other. But with the cytosis data, you can really appreciate that we are having more genes, and now we can really go into more detail to understand each of the cell type and cluster. For example, here, even though this is a gastric tumor, which they annotated as adenocarcinoma, we are finding genes that are actually likely representing neuroendocrine feature of these cell type here that are clustered in this group here. And we are also seeing the mucin producing cells from this region, and then the collagen secreting cells. That's probably fibroblast as well as macrophage that are in this cluster. And we also have the a more pure population of macrophage cluster that are representing in the green cluster here. For the AA2, again, because of the gene that are detected higher, now we are really seeing the separation of various cluster. We have the smooth muscle cell that are represented in this blue color here. And then we have the B cell plasma cell that are shown in this purple cluster here. And then these red area are the tumor cells. And we also have the mucin producing cells here and fibroblast secreting the collagen. So this is actually really powerful, and we are really pleased with the cytosis. And so we are now ready to provide the cytosis to our customer. Yep. So this is all I have for today. Thank you. If you have any question, you can type in the chat box. Does anyone have any question, maybe in the audience here?
It's great to see that the number of genes per spot has increased dramatically for version 1, version 2, but have you also seen the cluster number increase?
That's a good question. I think for the 251, perhaps we don't see increase of cluster because the data is already pretty good. Let me see here. I think cluster, there are 7 cluster, and I would say this is quite similar. Maybe just only one more cluster here. More spot, but I think it's because of the coverage of the tissue are larger. But with this 5 AA2, I think we have a much more cluster and more detailed separation. Yes. Mm-hmm.
One of the things that I've seen get folks really excited is it means this data just that you've shown, and now they're finding more of those clusters, even if it's in the gene, they're just finding more ways of taking momentum in different regions, right?
Yeah, yeah.
Reducing the size of the sample will affecting the result, because you say the strategies is a bit bigger than the
In this case, we are using the 6.5 by 6.5, so it's not changing more in terms of capture area, but the tissue itself, maybe I can show it here, right? Because this is the reg placement, and so we have to do it from freshly cut block. And so this one has been more exhausted, and this is actually the older section, like, it's more in the front, and so we actually have more tissue. So that's I don't think that is the case. We can actually find the same spot where we can then look at the number of genes detected. And even with this one as well, these are very similar area. But cytosis just have more genes per spot. Yeah.
I think you might have, maybe your question was more along the lines of you have options. So with the cytosis, you can choose 6.5, which you have 4, but you can also choose a bigger.
Yeah, and the area is bigger, and the strategies are affecting how much.
No, this is per spot, genes per spot. So it doesn't matter, it's, it's the average of the gene per spot.
Last year, slide.
2 slide, yeah, this one?
So at the bottom, you have increased the number of genes. How about the cell type? So the middle image is the active treatment.
Mm-hmm.
They have the cell type. And right one is the number of genes. How about the number of cell type? Do you have anything?
Yeah, Linda, so the data, this is actually not cells, it's spot. It's a cluster by spot. So yeah, it's not a cell type, so if we really wanted to know the cell type, we still need a single cell data to deconvolute, the result, yeah.
But it's good.
Yeah. Mm-hmm.
Very interesting. If you can do the same cell by spot, you have another layer.
Yes. Yes, so this is what Nassim's gonna talk about, yeah. Okay, if we don't have any more question, maybe we can then turn the podium to Nassim.
I try to share. Oh, yeah. Okay, I'm not mute, yeah. And that's okay. Thanks everyone for joining in today's webinar. My name is Nassim and I am a bioinformatics scientist here at ABio. Today, before going to the main topic of this talk, I'd like to spend a few slides introducing our bioinformatics services here in ABio. And then I go and talk about bioinformatic analysis on one of our research on human breast cancer using VCM spatial data. So in ABio, our focus is on single cell and providing multi-omic data from single cell, as well as the bioinformatic analysis on single cell multi-omic data. By using a single cell platform, we had made progress in treatment and diagnosis of many human-related diseases, such as cancer, cancer immunology, cardiovascular disease, and nervous system disease. Also, we have made progress in identifying new biomarkers for treatment and diagnosis. On the right, you see a bunch of papers that are published in this area by our team members. So our team members in ABio are experienced in providing different single cell data, such as cell surface protein, T cell receptors, B cell receptors, single cell transcriptomic, RNA transcriptomic, also VZM special data from fresh frozen and FFPE samples. And as my colleague Jasmine talked about recently, we have experience in using the recent 10x technique, cytosys, transferring tissue from glass slide to VZM slide and assessing gene expression from the VZM slide. About our bioinformatics services, we provide a wide spectrum of bioinformatics service, from very basic analysis to more advanced and customized analysis, such as RNA velocity, cell-to-cell interaction, and rare cell type annotation. And the table on the right shows in more detail what we do for bioinformatic analysis for different single cell data. We have very productive bioinformatic pipeline and computational infrastructure. Here is, for example, the single cell pipeline. We use it as a reference for other type of data. Our team in ABio not only provide bioinformatic pipelines, we also have developed new bioinformatic tools, new algorithm to answer new scientific questions. For example, all the tools that you see here are the ones that are developed by our bioinformatic team. StarTrack is for tracking the dynamic of the single cell, CSOMAP for cell-to-cell interaction. Cybet is a fast Bayesian method for cell type annotation, and Roche is for identifying the heterogeneity of the tissue. Today, I'll be talking about our bioinformatic analysis, integrating 10x FFPE VZM and a public single cell dataset to reveal the cell type composition, spatial information, and malignant cell state of human tumor samples. Just to remind you about the importance of the spatial data, we are all familiar with the heterogeneity of and complexity of the tumor microenvironment. So tumor microenvironment composed of different kinds of cells, lymphocytes, cancer-associated fibroblasts, and tumor cells. Using bulk RNA to understand the tumor biology is not very helpful because we get mixture signal from different kinds of cells. Single cell, to some extent, resolve this problem, heterogeneity problem, but it doesn't provide any information about the location of the cells. On the other hand, the spatial data, we can get gene expression from each location. In our study design, we had 2 samples from human breast cancer. And after assessing the RNA and a slide preparation, we move forward into FFPE gene expression assay and provided library, and those were sent out for sequencing. Today, I'll be talking about the bioinformatic analysis of those VZM spatial data. Just give you a brief overview of what we did in bioinformatics. After looking at the data quality, we performed an unsupervised machine learning method to identify the clusters in the VZM to understand how many different cell types we had in our tissue. And next step, we use a supervised learning method to understand each spot or deconvolute the spots. First step, the reads of VZM spatial transcriptome mapped back to human transcriptome to provide the spot level UMI or spot level gene expression. And those information were superimposed or overlaid on the real tissue. And that's what you're looking here. The 2 samples had good quality. Sample number 2 on the right had a slightly better quality. You see most of the spots are in red region or highly expressed region. So for downstream analysis, we selected sample number 2. And here is the 10x Space Ranger standard output of sample 2. And some important criteria to consider for the data quality is, for example, the number of reads, valid barcodes, valid UMI, median genes per spot, and median UMI per spot. On the right side, you see the basic analysis by 10x loop browser, in which the spots were clustered into 8 different groups. Well, after looking at the data quality, we move forward with our bioinformatic pipeline. So the count matrix and the UMI matrix converted to a Surat object, and we use the Surat R package for downstream analysis, for doing normalization, dimensionality reduction, or PCA, clustering, and cluster annotation. And you see on the right, the UMAP from Surat clustering result, clustered them into 14 different groups. The next step, we perform a spot deconvolution. So as you know, in Visium, each spot is not exactly one cell. It's composed of 5 to 10 different cells. And by a spot deconvolution, we can infer how many cells and how many different cell types are exist within each capture location. To do that, we use a Spotlight. There are different computational tools to do that, and we use a Spotlight. It requires that we give it a well-annotated single cell data reference as a reference to be trained by the bioinformatic tool. Because we didn't provide the single cell data from the same Visium tissue, we use other resources, other studies. There was this study in 2021 in Nature Genetic, in which they provided a single cell reference for human breast cancer. And that single cell reference actually was integrated across 26 primary breast tumor and included 130,000 number of single cells. From that single cell data reference, as you see in the UMAP, we see 9 different group of cells like B cells, cycling cells, endothelial, epithelial, myeloid, and mesenchymal cells that included endothelial, PVL, perivascular-like elements, and cancer-associated fibroblasts. And those clusters were annotated based on the expression of canonical gene markers on the top right. So using this single cell for the convolution requires some pre-processing to make it more computationally efficient. Like for example, we selected the highly variable genes around 3,000, and then we downsampled the data to 900. For each 9 cluster, we selected 100 of cells. Before performing this Spotlight, we also look at the expression of some canonical markers in our Visium data. For example, BANK for B cells, CD3T for T cells, MS4A6A, and CD68 for myeloid cells, EpCAM for epithelial, and so on. So we can see by just looking at the expression of canonical markers on the Visium data, we can see the heterogeneity of the tissue, and those regions are exactly especially distinct. Going back to Spotlight for deconvolution, so as I said, the Spotlight requires a well-annotated single cell data as a reference to deconvolute the Visium spots. And it works based on 2 models, 2 algorithms. On the right, that's the figure from the Spotlight paper exactly, and it shows how step by step it works. In general, in one step, it defines some topic profiles, cell topic profiles based on the gene expression signature from the single cell. And the next step, it actually optimizes the proportion of different cell types that fit within each spot. By a Spotlight, we could deconvolute our Visium data. On the left, you see the real image. On the right, you see the special scatter pie representing the different kind of cells from the reference, single cell reference in our Visium data. And if we zoom in each spot, we see each spot is a pie chart showing different kind of cells and different proportion of cells within each spot. For example, here that we see more of pink color is the region that mostly occupied by cancer epithelial cells or the tumor cells, breast tumor cells. The score that we get from a spotlight for each cell type can also be used to identify the location of the cells on the Visium. For example, these two regions are the region that we got highest score for B cells. Here is the region that we got highest score for T cells. It seems myeloid cells are distributed across the tissue. Here in lower part is the region that we got highest score for plasmablasts. This is the image that I showed before, is the region, the ribbon shape, is the region that we have highest score for cancer epithelial cells. And cancer-associated fibroblasts are located on the upper part and lower part of the tissue. We also use another approach to identify the malignant cells or tumor cells by inverse CNV analysis. So the study shows that the human breast tumor usually derived from changes in DNA copy number. And that's actually what the inverse CNV analysis does. It can estimate the copy number variant by looking at the relative gene expression intensity in the tumor cells and compare it with normal cells. So we selected two of clusters, cluster number 1 and 7, in which we were sure there is no tumor cells as a reference to use for inverse CNV. So the inverse CNV heat map on the top, you see the relative gene expression across the chromosome in the reference. And the lower part, you see the malignant cells versus non-malignant cells are separated from each other. Malignant cell, for example, we see higher intensity expression in chromosomes 7 and 8. And those are the regions that probably we have the tumor spots. The in situ visualization of the malignant spots also aligns well with the region that we got highest score for the cancer epithelial cells from a spotlight, and this is from the inverse CNV, and also aligns well that we get highest score for cancer-associated fibroblasts in lower part. We also integrated histology to our study. We had pathologist annotation ahead, and eventually we compared the pathologist annotation with our spotlight annotation. On the right is the pathologist annotation. The blue regions are TIL or tumor-infiltrated lymphocyte. Those blue regions align well with the region that we got highest score for B cells, T cells, and myeloid cells. On the left, you see the fibrosis, the green region annotated by pathologists. They also align well with the region that we got highest score for cancer-associated fibroblasts from a spotlight. The same for epithelial cells. And the vessel elements are these purple regions that were annotated by the pathologists. These also match well with the region that we got highest score for PVL or prevascular-like elements from the single cell. One of the cell types or tissue that we would expect to see in the breast tissue is the adipocytes or fat cells. That annotation was missing from our spotlight because the single cell that we used for the convolution didn't have any fat cells. And that's because how they dissociate the tissue and RNA capsulation. Instead, we look at the expression of one gene markers for fat cells, which is fatty acid binding protein 4. And we see the expression of those gene markers align well with the region that were annotated as fat tissue from the pathologist. In summary, we use 10x BCM technology combined with our deep analysis technique to explore gene expression characteristic of FFP human cancer tissue. By using comprehensive single cell reference data as a reference, we could build a precise map of cell type annotation within each spot, interrogated the regional occupation and co-localization status of different cell types. By looking at copy number variant inverse CNV analysis, we could identify the malignant cells versus non-malignant cells. And finally, in all cases, our pathologies annotation correlated well with our cell type prediction and gene expression annotation. Thanks for listening. And if there is any question, I'd be happy to take your questions. Yeah.
That was really beautiful work. Would you ever do analysis on samples outside of your group having done the initial wet lab work? Like, let's say a group had, let's say, a mountain of data that they've accumulated. Would your group ever take that upon themselves to sort of like do analysis and sort of like what you've done here, even though you didn't do the front work part of it?
Was it this one? You guys prepared the lab work?
This one we did, yeah. But I think they are quite a few that is,
Yeah.
team actually do analysis.
Yeah, we do only bioinformatics.
I know that's folks talk about the wet lab work being the bottleneck, but once you get that amount of data, that could be a bottleneck itself too. I guess.
Exactly, yeah. So we have those as well.
Oh, okay.
You see, my mouse doesn't work. Do you see any question in the chat?
No, I'm not sure. So this actually data is coming from ECV1, and the number of genes per spot is actually already very good. It's 6,000 genes per spot. And I think we can get another data. Even with the previous version that we used. Yes.
So what are the copies? Okay.
Maybe copy of the probe, only one copy.
So this is one single mRNA with a hyperlap is one.
Yeah, supposedly. Yeah, I mean, the probe is designed by 10x, and the RNA is fragmented, right? So it's only one conserved region that was copied by this one probe. You might miss it, but like in our case, I think we detect the majority of it. But if you have a more degraded RNA, maybe you will miss it. So with 3 probe per gene, then you have more chance to detect that gene. Yeah.
For version 2, though, we have 3 probes. 3 of the average of 3 probes. So 3 different probes.
Yeah, using averages like 3 probes per gene. Yeah, for better coverage and protection.
Which helps with all that fragmentation, right? So gives a better chance of getting in there and finding the target for fragmentation.
Where do you put like all 6,000?
Well, that's just what was detected per spot. So it covers 18,000, which including transfer.
I see. So, but you have some selection, and increasing the purpose. If you only just randomly select
It's just, it covers the whole protein coding transcriptome, so around 18,000 genes is covered, and then just in any region that you're detecting gene expression or in the experiment, you have those individual spots, and so from a median like genes per spot perspective, you're getting about 6,000 genes detected.
So the final goal is to cover all of it.
I mean, it does, so if there was, you know, another spot over could have like a different selection of genes that were expressed in that part of the tissue.
The overarching goal was to give the highest.
So that you mean like given a spot, you have a given set.
No.
Same set.
The same set, but we're only detecting what's biologically relevant there or present, right? But in terms of coverage, you know, the group did speak to key opinion leaders and ask, what do we really need there? We want obviously the biggest coverage, but we didn't want it to be a burden on the experiments. We actually did remove a lot of things that were necessarily having fun, things like that. So the 18,000 that were chosen were what 10x felt was the highest span coverage of targets that we could see in the sample.
I mean, so it doesn't cover, I think, like the MHC genes and then like TCR, very variable genes. So, you know, it's again, it covers by and large the whole protein coding transcriptome. And then as the gene expression patterns change throughout the tissue, you're just going to detect different gene expression from that, you know.
But since you said, right now it covers 6,000 genes.
No, that's only in our experiment.
That is in that particular.
That's in that particular experiment of the breast tumor.
Okay.
And each spot average is 6,000.
Sample.
From that sample, 6,000 per spot. And imagine how many cells is in that spot. Up to 10 cells, for example. So the 10 cells are giving you a signal, you know, from it. So each cell maybe 2,000 genes. There's going to be overlap of genes. So in total, from that spot, average is 6,000 genes detected.
It's a reflection of the cells that are there. Like I've seen data from the PBMC where they're very, you know, rudimentary cells, so there's a smaller number of genes there, right?
What are the difference in sensitivity V1 versus V2 in terms of the lowest limit of the mRNA?
That's a great question. No, I mean, we've seen example. I mean, that's one of our selling points. We've seen examples now talking to customers that the sensitivity is higher. We've seen people adopt this specifically for the probe set being a higher sensitivity because you're targeting instead of going for the sequencing path of getting everything. Right? So we have folks that have adopted this specifically for the sensitivity, knowing that a probe-based approach is a more sensitive approach. Yeah.
Do we know the limit of sensitivity?
I think the limit would be a target, a molecule. I don't think it's limited like more than that. You know, I don't know if you're going to find it every time, but I think that is the limit.
Yeah, I mean, I don't know if I have a great answer for you, but I will say that so we have a single cell application, and just referred to it with our, so you can dissociate an FFPE tissue or use fixed samples, and it uses the same probe set essentially. And we've done a lot of comparisons between just a reverse transcription based single cell methods like 3' and 5' with this probe-based detection. And so, for example, there's a preprint out there that ran like a side-by-side comparison with our fixed RNA profiling or single cell gene expression flex versus, you know, with our 3' single cell gene expression solution. And so they actually point out that they are able to detect some of the lower expressing genes like transcription factors a little bit better with, you know, the flex probe-based method versus reverse transcription. And that even though they're detecting the same clusters of cells, that the clustering is a little cleaner when you're able to pick up more genes per cell, or in this case, per spot.
For single cell or non-spatial reverse transcription base, what was the limit? Is one molecule or what is it known?
I don't know. Yeah, Yeah,
Either.
yeah.
Yeah.
Theoretically, I think both should be able to give you the molecules, just how often in practice would you see that?
Yeah.
And just the data that we've seen is showing that, well, with this method with the probes, this is when the recurrence will be higher for finding, right?
Is there such thing as an upper limit? Like, in theory, could it be more RNA molecules than the probes?
That's a good question.
Can you repeat so people online can hear? I'm getting messages that can't hear the whole conversation very well.
So,
You can be heard. So you must have your speakers on.
oh, I can't, I'm not even joining. I haven't
Okay,
joined.
yeah, so the question is that is there upper limit of the RNA molecule, you know, meaning there are more transcript in the tissue than the probe itself. Right,
Oh, man, this takes me back to my other days of probe-based chemistries. I can't answer that. I don't know, but from my previous experience, I don't think any probe, any target that you would have would hit the upper limit of the reagents that we have in there. I think
Yeah.
the sheer amount of molecules that we have in there, the enzyme or terrible answer. I don't think so, but I'll find an answer for
I
you.
was thinking like in terms of how many primer probes you put in there, like is it hundreds, thousands per single gene?
Yeah, I don't know,
No.
you
I'm
know.
just curious.
Yeah,
Yeah.
but I would imagine that it's probably not like saturating in terms of the RNA transcripts that are there. Again, I think that also comes back to just the comparisons that we've done with like the reverse transcription based methods versus the probe based methods with the single cell application. And we can certainly, in many of the sample types that we've tested, we see more genes per cell with the probe-based methodology. Yeah.
I think given how many people are using this now, that's probably an answer that we can get pretty fast, just asking, what type of tissue makes a whole lot of something and see if we see some sort of saturation marker or number, right? Yeah.
But yeah, we do have a lot of data sets for Visium on the website. John brought it up during his presentation. And so there's very diverse tissues that are on there, and some of them might have more dense regions that you might expect more genes to be detected or RNA transcripts. And so that's certainly something that you can take a look at and maybe see how, the range that you sort of pick up.
I think that's why it's not a hard answer in terms of how many probes we have in coverage. It's an average, because for some high expressors, the number is lower, like for actin and things like that, cytoskeletal ones where there's a lot.
Yeah,
Yeah.
and that information is available. And so there is like a probe list, and it'll tell you like how many probes for this gene, for example, on our website. So you can download it. I may have missed this, but in your presentation, when you're using something like Spotlight for the single cell deconvolution of the Visium spots, is it important to have single cell data that represents that sample, or are you able to use like reference data?
Well, ideally, yes, if you can get the single cell data from the same Visium tissue, that's the best thing, but at that time, that was not possible. We used the most comprehensive thing that we could find.
Okay,
And it worked well.
thank you.
Actually, you'll see the sensitivity difference between
Like the fresh frozen versus FFPE.
Yeah, so in terms of the, like, side by side, we don't really compare it in a way, that's like a side by side direct comparison. We don't track that data in-house, but what we did was, in terms of what type of sensitivity we can obtain, in a way our goal was to match the sensitivity we were seeing with fresh frozen. We ran the fresh frozen assay. We could look for how many genes per spot could be detected with that. That's of course direct poly-A capture as opposed to working with probes. So there'll be a difference there. So it's not like comparing apples to apples there. It's not direct, but in terms of the sensitivity using those two methods, we do see similar sensitivity. We're able to get, but it's still, you're working with a different approach, right? So it's not a direct comparison. But in the number of genes we can detect is relatively the same. We do get to a very similar spot, and I do have a graph I could share. I can share it after the meeting. It shows how we ran an FFP section on the fresh frozen assay before FFP, the assay was available, and then later we introduced the probes. We were able to get near the same sensitivity, but in a different way, right? Because we're using the probes instead of polyethylene.
So maybe a little bit of stretching. Because cancer samples is so limited, not every cancer patient could have the tissue samples. So have you guys done any like correlation, see the tissue and then compare with the periphery, like blood liquid sample, see whether you can have some correlation there? So for example, like pancreatic cancer patients, maybe 100 of them, maybe only 10 have tissue samples. Then you have all the signature, but then the rest only have blood samples.
That's a wonderful idea, almost like doing like a liquid biopsy, given that you already have the signature from a true biopsy, not from the same person because they're still alive.
Right.
I can't answer that right now, but I think I'm sure there's got to be people working on it. We'll ask around. But that's the holy grail right there, right?
Have your clients or anybody asked this type of question? Has this need?
The customers that I have, no one's asked that explicitly. I can't imagine why why Wolves wouldn't be doing that, because that would like,
Ideal.
ideal, but I don't think anybody wants to reveal what they're doing, But I think you're hitting about something really amazing, because as soon as we reveal that you can take, blocks, FFPE blocks,
Right.
and get single cells from them the way that Jasmine has been doing, every single large pharma that I know of started doing that. And I think it's because of that. They want to develop that signature and then correlate it back to a liquid biopsy that they don't have to actually take anything from someone, right? Yeah.
And it's getting harder, the situation for those cancer patients who become resistant. And you probably get some tissue.
Exactly. So you can see the
really
dynamic
fast.
nature of the tumor. Right?
Exactly.
Exactly. Yeah.
But your technology is not designed to handle liquid biopsy.
Depends on what you would consider liquid biopsy, right? I mean, you could take blood from a mouse or from a human and get single cells. It wouldn't be the spatial, but we could definitely get single cell data from that.
But that's not a spatial pathology, biology.
Right now.
Yeah,
Just single cell.
that can be handled by our technology.
What our single cell offering you.
Yeah, I mean, I think it just depends on what you're looking to get from that sample, and so there's many measurements that you can get from a single cell suspension. And so we didn't talk about it today, but things like looking at TCR or BCR clonotype analysis from the immune cells that are in there. You can look at cell surface protein measurements from the cells that are in there, gene expression, all of those types of measurements.
We do a lot of single cell PBMC
Yeah.
studies. Yeah, you don't have to be spatial to do
Exactly.
10x technologies.
It's like that's all.
Yeah, so thank you very much for joining us today. And we thank 10x Genomics and John for presenting, and thank myself. And thank Nassim for the presentation. Okay, bye guys. Thank you.
Thank you, Jasmine and Eva, for having us here and for this event. No, we really appreciate it. Great.
Very nice experience for us.